I chose to create this problem because I think it really puts in perspective the genetics behind amniocentesis in a way that isn't too complex. It helps patients understand what's going on behind the scenes of the procedure they went through and how it is being used to prenatally diagnose their child. So the problem starts off using strict genetics, where we see things like these DNA sequences being used as probes and the results of FISH analysis seen here in the form of a cell with a bunch of chromosomes. But after recognizing what these findings show, it becomes more personal again. The problem is asking to take the results of a genetics problem and use them to diagnose. It applies science back to the everyday life that non-scientists are familiar with, which is important in the context of bioethics. So let's actually dive into this problem. So we see you are 22 weeks pregnant. You have a history of multiple failed pregnancies, so your doctor recommended amniocentesis to assess the chromosomal occurrence of your fetus. You agree to the procedure. Because you have an interest in genetics and some background knowledge in the field, you ask to receive any preliminary findings as soon as they result. Your doctor provides you with the following report and a photo of one of the FISH studies. So looking at the report, the first thing that we see on the report is that the findings are going to be representative of chromosomes 13 and 21. This is important because knowing which chromosome has abnormalities is important for diagnosis. The probes used are listed below it, as seen right here. We see the sequence of the probe 13. This means that this is a sequence that is complementary to chromosome 13. Assessing the probe in testing means that it should attach to a sequence within chromosome 13. And then when we visualize the results, anything that's highlighted in blue is going to be representative of the presence of chromosome 13. The same thing goes for probe 21. The sequence we see here is complementary to a segment of DNA seen in chromosome 21. So any visualization of green fluorescence is going to indicate chromosome 21. Okay, so now that we understand that, we can look at the FISH study results. Let me scroll down. We see a bunch of chromosomes in a cell with multiple colors. Remember, we said that anything colored in bright blue is going to represent chromosome 13, while anything in green is representative of chromosome 21. Based on what we know about humans, there should only be 2 copies of each chromosome, which adds up to a total of 46 chromosomes. Well, we see that we have 2 green chromosomes. Here's 1, and here's 2. This is normal, which is good. Then we see that there are actually 3 copies of a bright blue chromosome. There's 1, 2, and 3. This means that there are 3 copies of chromosome 13, which is abnormal. Having 3 copies of a chromosome is indicative of trisomy. Using this information, we can scroll back up to table 2.1, which I posted here. Let's go down this list and find a condition consistent with what we just visualized. So first we see polyploidy. The first condition is triploid, which means that the individual affected has 3 copies of every single gene for a total or 3 copies of every single chromosome, for a total of 69 chromosomes. This is not consistent with what we just saw in our FISH study. The second type of condition is trisomy 13. This means that there are a total of 47 chromosomes seen in the study, with chromosome 13 having an additional chromosome for a total of 3 chromosomes. Now, if you remember, this actually is consistent with what we just saw in this FISH study. We saw that there were 3 blue chromosomes, and we said that blue fluorescence represents probe 13 for chromosome 13. And this means that the fetus is likely going to have Patau syndrome. We see that there are other conditions that are similar to that. So if the study would have shown 3 copies of chromosome 18 or 3 copies of chromosome 21, then we could have diagnosed with Edwards syndrome or Down syndrome accordingly.